My name is Jacob Schreiber, and I'm a postdoc in the genetics department at Stanford. Go fighting trees. And so today I'm going to introduce you to a project we've been working on called TF-MoDisco-lite, which is an attribution-based motif discovery algorithm. But I'm not just going to talk about that. Rather, what I'm going to do, because I think there may be a diversity of backgrounds in the room, is that I'm going to introduce you to a central question in genomics. I'm going to talk about old methods for trying to approach this. I'm going to talk about what happened when deep learning hit the field, and then how TF-MoDisco fits into that new paradigm. And then towards the end of the talk, I'm going to describe why this is TF-MoDisco-lite as opposed to just TF-MoDisco, and I'm going to show you some how to use it, basically. And the reason for this is pretty simple. It's that using the concept behind TF-MoDisco is pretty simple, and how to use it is pretty simple. But what's difficult is setting up the right problem. And it's easy to mess up how you set the problem up and to get results that aren't particularly meaningful. So without further ado, a central question in genomics is why do we have so many cell types given that every cell has the same genome in it? You can see here in this cartoon that there are a whole bunch of different primary tissues that are in the body. In addition to all the ones that are pointing the places on the body, our brain is made up of all types of very distinct types of tissues. Our skin are made up of various different types of tissue layers here. But there's more, that we have cells at different stages of differentiation, that we have our stem cells at the beginning, and they give rise not only to the fully differentiated cells, but a whole host of partially differentiated and transient cell states and all types of really exciting things. Finally, there are cells at different stages of development, that fetal versions of tissues can be very distinct from their adult counterparts. All of these cells have basically the same copy of the human genome in it. So what's happening? Well, for many of you know the answer to this, which is that even though they have the same copy of the genome, that the genes that are encoded in the genome are expressed at different combinatorial levels, and that's what gives rise to those different cellular phenotypes. That basically the different sets of genes will be on, and that turns the cells into different things. But that's not the full story. That we can say, okay, gene expression is this way in this cell and another way in another cell, but we don't know why. We don't know what gave rise to that, what the consequences of it are, and really what are the driving genes that are causing the cell to assume that state. Because there can be many types of changes to gene expression that don't really matter, but there are some that do. And so the way that we look at this is we look at various types of cell type specific assays. RNA-seq being one that measures transcript abundance, but also we have things like DNA-seq, FAIR-seq, ATAC-seq. These measure general chromatin accessibility, which is like if the genome is more accessible to the cell, stuff is probably happening there, and so that's going to be a good place to look. What we're going to focus on in this talk is ChIP-seq. This is a type of assay that can measure protein binding along the genome. And so looking at these assays just by itself can be really informative. If we look here at this locus, this is the NANOG locus. So NANOG is a key gene that drives cellular reprogramming. But we can see that there are a whole bunch of other genes nearby. Interestingly, if we look at CTCF is kind of an outlier protein, that it's doing its own thing. It's this big, bulky protein that binds to the genome, and it just kind of sticks there. It doesn't really have cell type specific activity, but when it does, that's incredibly important. And it basically just prevents anything from happening in that region. But more importantly, it coordinates the 3D structure of the genome. Basically, it helps make the genome fold in the way that matters. And so here, what you can see is that we kind of have this box around NANOG. That NANOG is interacting with the genes that are around it because CTCF is forming this little loop, basically that's called a topological domain, where all of the genes inside it have a similar regulatory environment and talk to each other more. So by looking at this single assay, we're able to get a lot more insight into what's happening at this locus than we could just by looking at gene expression values. Interestingly, though, there's kind of this middle peak here, which is transient. In some cell states, you see that, and in others, you don't. And this is important because there's a set of genes on the left-hand side of it that sometimes interact with that NANOG and the NANOG genes, and sometimes don't. And that can be important in driving cell fate. And so in this case, because CTCF is present, it acts as a blocker preventing NANOG from basically talking to those genes. So we have all types of complementary information here. But before we go on a little bit further, I have to look at this. I have to look at this locus a lot because a lot of my research involves cellular reprogramming, and I think that, like, gene naming is really weird. So we have nanog, right? Nanog derives from tir nanog, which is the iris spring of eternal youth, and that's a really cool name for a gene that's involved with reprogramming because we're trying to use reprogramming to make things, youthful. If you look right next to it, right here, there's this gene nanog NB. That literally stands for nanog's neighbor. And that's like aggressively mid for a gene name, right? Like, if you're going to put so much effort into nanog, you should at least give equal thought into its neighbors. So coming back to this, the questions that we're going to be focused on are what gives rise to this CTCF binding here? Like, obviously it has to do something with the genome, right? But what is it specifically that's causing CTCF to bind at these locations and not other locations? So before we get into that, though, the main question is, why do we care? Like, if we have these assays that can tell you where CTCF binds along the genome, why do we care about understanding the underlying genomic basis of it? And there are kind of three reasons. The first is we're advancing basic science. The more we understand, the more enlightened we are as a people. And that's kind of a boring reason. The next one is that it's actually quite useful for medicine. We're starting to get into an era of personalized medicine. And so this involves basically trying to sequence your individualized genome, find where your mutations are compared to reference, and see what the effect of those mutations are going to be. You can imagine that if you had mutations in a CTCF site and you knocked out CTCF binding there, that's probably going to alter the 3D structure of the genome. And just like how that seemed to matter for nanog, you cause genes to talk to each other that shouldn't, and that can be an underlying cause of disease. So this is very useful in screening for people to see whether or not they're predisposed to developing illness later on. The second is that with the advent of CRISPR, we've been developing a lot more therapeutics that are based on engineering sequences, that either we want to try to revert bad mutations in the genome, or we want to create some construct that does something useful in the body. And so the better that we understand the sequence basis of various things going on in the genome, the better we can construct these things and make profit in our capitalistic system. So let's return here. The way that the methods worked in the past is that you'd find the peaks of the thing that you cared about. So this might be like promoters for genes, or if you had a ChIP-seq assay, you'd do peak calling. You'd identify these peaks, and you'd extract the sequence under them. The idea would be that if this is really caused by something genomic, that after we extract the sequences, we can then look and find the pattern. I'm sure all of you have already found the pattern here, but there are machine learning methods that try to scale this up to tens, hundreds of thousands of sequences. Some of the first approaches involved doing multiple sequence alignment. The idea there was basically that you take all of these sequences and you would try to align them to each other, and they would align based on the conserved part of the sequence. And so you can see that the inner part is CATAA. That is the common motif that is in all of these sequences. And the idea would be that if there are common motifs here, that is what drives the sequence. The problem is this is incredibly slow. That multiple sequence alignment in its exact form is NP difficult, and there are heuristics, but each one of them are still very slow. And so there are more recent approaches like MEME, which try to phrase this as a mixture model. The idea is that you want to try to separate you want to try to cluster k-mers into those that represent protein binding and those which are sort of background. And so you take each one of your sequences, you slice it into these k-mers, and then you run basically this classic mixture modeling on it, and you're able to pull out the repeated parts of the sequence. So regardless of this approach, because those are two, there are many others, you end up with something called a position weight matrix. And so for each position in this motif, it's saying basically how conserved is each nucleotide. So this is the PWM for Vest, which we're going to return to later. But you can see that there are some nucleotides where it matters a great deal that you have an exact match there. But there are other nucleotides that are more, they can be one of many. They don't seem to matter as much. In particular, for this PWM, it seems to say that there are two nucleotides right in the middle there that don't really matter. And it's mostly the flanks around it. This is going to be interesting, and we're going to look at it later. But this is basically what the state of the art in the field was. So More recently, with the advent of deep learning, the idea here is that rather than using the ChIP-seq experiment simply to identify peaks and then extract sequence from under it, what you do is you train a machine learning model in order to take in sequence and predict the readout of that experiment. And so the idea here is that you are trying to predict the ChIP-seq experiment using whatever machine learning model. In our lab, we use one called BPNet, and you can see that for this example I'm showing you that the predictions end up being quite accurate. We can see here that for one of the motifs that you have the observed signal on the top and the predicted signal below. And then for a whole bunch of other ones, you can see it's basically doing the right thing. An issue here, an issue in genomics in general, is that prediction accuracy doesn't really matter that much other than showing that your model works reasonably well. There isn't more secret genome that you need to make predictions for. When you train a machine learning model in this way, you are training on data that encompasses the whole genome already. And so the real purpose of these models is to then backtrack to say, what is it that you learned? And so the real focus here is on the feature attribution methods that you subsequently use. In our lab, we use one called DeepLift, but for the purposes of TF-MoDisco, it doesn't really matter which one you end up using. So if you make predictions at this locus for these 4 different TFs, you can see that the predictions seem reasonable. But if you backtrack down to the sequence, you can identify the sequence elements that are driving those predictions. You can see that commonly all 4 of these seem to care about the OX-OX locus, which is what's coordinating the binding of all of these proteins. But that additionally, NANOG in yellow seems to have this additional NANOG binding site that's nearby. And KLF4 is flanked by those high G regions, which are the classic KLF4 binding sites. So this is incredibly powerful because this is really fine mapping the signals that we were getting before. Instead of getting peaks that are 100 or 200 base pairs long, we are getting the exact nucleotides that we think are driving the signal. And this is what TF-MoDisco is going to be operating on. So finally, we get to TF-MoDisco. This idea here is that we want to identify these repeating patterns that we're finding, because if you can just, if you start off and you do attributions, you can see what happens at each locus, but we want to see what's happening more genome-wide. So what you put in are the attributions that you get in addition to the sequence. The first step is to identify these things called seclets. You basically want to chop this array that has the importance into short spans of high attribution regions. These are basically what you anticipate the motifs are going to be. And after you do that, you then divide these into the positive attribution seclets and the negative attribution seclets, because this is very important biologically. There are motifs that are going to be driving the thing you care about, and there are ones that are going to be repressing it. And so you want to know, you don't want to just average those out and either get no effect or have a diminished effect on one side. If there's a motif that's both driving and repressing, that's very interesting, and you're going to want to look at it later. Then, because what you want to do next is cluster these seclets into the ones that are similar to each other, but a problem is that we can't store that in memory because there are going to be thousands, hundreds of thousands of seclets, and we can't store a 500,000 by 500,000 similarity matrix. So for the purpose of time, I'm just going to skim over this and say that the first step is to calculate a coarse-grained representation where we just keep the top, the nearest neighbors that we anticipate are going to be similar. Then on that sparse matrix, we refine those scores with a high resolution score that we actually care about, which involves doing alignment of all the seclets against all the other seclets. We do a quick density adaptation thing, and then finally we cluster. It's really conceptually simple. The first step is pull out the seclets from your sequence. The second step is basically cluster them. And by doing this clustering, you can then pull out the repeating patterns that are the things that are driving the sequence you care about. And what you end up with is a report that looks something like this. Each line is the aggregate motif that was found. Basically, it's a pattern that was found repeatedly. We have the sequence on the forward strand, then on the backward strand. And then if you provide a motif database that you'd like to compare against for annotation, then it finds the best hit in that database. This is usually just so that you as a person can get a gist for what does TGA, TCA mean? What is that probably associated with, right? Okay. So I want to make sure that we get through these case studies. In the first one, because these reports can be extremely useful in identifying what's going on. So we started off and we trained 2 models, one that predicted E2F6 binding and one that predicted max binding. When we did the whole pipeline of we train the model, we do the attributions, we do the TF-MoDisco, we found that when we looked at the E2F6 model, that we found both the max motif as well as the E2F6 motif in it. In contrast, when we looked at the max model, we only saw the max motif. This suggested some sort of asymmetric effect was going on, where E2F6 might be influenced by both of the motifs, whereas max didn't really care about the other one. And so when we went and looked at the actual genomic data, parts of the genome, this is what we found. This region here on the top is the actual observed signal at some locus on the genome for E2F6 experimental data. And then we have the experimental data, and then we have the predicted data under it, and then we have the attributions. And the attributions here are highlighting that for E2F6 binding, it cares about both of them. But for max binding at the same location, it only cares about the max motif, and it's much more shifted to the left. If we just look at overlap of peaks, we can see that E2F6 peaks highly overlap with max peaks, whereas there are a whole bunch of max peaks where there's no E2F6. And finally, we can just do a perturbation experiment where we take a sequence that doesn't have any binding of either of them. We can add in the max motif, and we see that we get max binding from both of them. We can add in the E2F6 motif, and we see that we don't really see any max binding from the max model, but we do from the E2F6 model. And so this is really cool, because cofactors are kind of an understudied area in genomics. And this just very cleanly showed from the TF-MoDISco report that there was this asymmetric effect going on. And that, that can lead to a whole range of automated ways of pulling out these types of cofactors without the need for doing these types of complicated analyses. The second one is returning back to rest. If we look at the top, this is the motif that we were seeing before. It's basically the canonical rest motif. Rest is one of these zinc fingers, has a whole bunch of fingers. Each finger binds to 3 to 4 nucleotides, but there can be different spacing constraints. And this is something a PWM cannot capture, that if you don't care about the exact number of base pairs between the fingers, there can be an additional one, you can't have a rigid static PWM. And so what TF-MoDISco pulled out is that there's actually a left half of this zinc finger and a right half. And it said that something like this can happen where there's a whole bunch of spacing between these two elements. This is just something that fell out of the TF-MoDISco report. And so we were able to find a whole bunch of examples of this along the genome. Yes, we found canonical examples where there's only that 2 base pair spacing between the left and right half, like you saw in the PWM. But there are also a ton of examples where the spacing is much larger. And this makes sense biologically because if you have the zinc finger protein, a PWM is not going to capture it well, and yet we keep insisting on using PWMs to represent all of our data. I think I'm sort of running out of time. So I think suffice it to say that originally the implementation of TF-MoDISco was very research-based. It was written by a researcher, and so it ended up using 800 gigabytes of RAM, which was a little bit too much for people. And so here, this is a more reduced example. I was going to give some advice here, but suffice it to say, look through your code and use a memory profiler, because there was this spike here that used around 20 gigabytes of memory, and that was due to this one line that seemed fairly innocuous. By pinpointing that line and getting rid of it, you were able to get this fixed. And by the end of the whole procedure, I think I'd removed like 10,000 lines of code, significantly reduced the number of files, and we got it down from, in this example, using 80 gigabytes down to using under 8 gigs, and around 2 to 3 times faster. So using it, there are basically 2 steps. The first is that you use this command line tool, MoDISco, which comes with the MoDisco-lite repo that you can pip install. And you just do MoDISco motifs. You pass in your one-hot encoded sequence. You pass in your attributions from whatever algorithm, deep lift, ISM, whatever you want to do. You give it the number of secrets you'd like to use, which should be as large as memory allows. This was largely an artifact of used to taking 800 gigabytes of RAM. And then you get the results back. What you get is an H5 file with all the patterns, and for each pattern you get all the secrets that are mapping to it and a whole bunch of information about it. H5s are kind of unwieldy, but there's still a universal data type that you can be loaded everything, and it's useful to have all of this information around. And so finally, there's a command for generating the report. It basically just takes in that H5 file, you give it a path where you want to save everything, and if you want to have a database where you cross-reference it, you can pass in that meme file. And when you do that, you get this report. So I want to thank all the people who are involved in this project, all the various co-authors that have been around since the beginning, as well as the brains behind the operation here at the end. So, thank you, and I'd be happy to answer any questions.
Forgot I did the microphone thing. Testing. Yeah, that was great. Thank you. So it looks like you got you're training two neural nets together to do this. And I was curious about the first one, when you're saying backtracking. Are you just like ripping out a latent vector and then?
Sorry, there's only one neural network here. This is one of the strengths of this new approach. The idea is that you train a neural network to predict chip-seq signal. And then with that same neural network, you just interpret it. And so these methods that basically, for a given input, you make a prediction, and you can explain what about the input, like why did it make the prediction. So there's no like latent vector, there's no, yeah.
Okay, so are you using perturbation to light up is that your method of interpret?
Yeah, so TF-MoDisco-lite is agnostic to the attribution and the approach you use and the model approach you use. In our lab, what we commonly use is this thing called deep lift, which is a modification of gradients. And so it ends up being a lot faster because with perturbations, you have to run one perturbation for each nucleotide and each possible nucleotide. With gradients, it's just one backward pass. And so it's quite fast and a pretty good approximation of ISM.
Awesome, thank you.
Thank you. It's a great talk. I'm Carol with the University of Colorado. Just curious, have you tried this with ATAC-seq?
Yes.
Okay.
So that's a little bit more complicated.
Yes.
Sorry. So on the TF-MoDisco side, it's just as easy. The problem is that with ATAC-seq, there's this TN5 bias that convolves the signal. And so if you try training a machine learning model directly on ATAC-seq signal in the same way that we are training on chip-seq signal, you're going to get the TN5 bias. Everything's going to be messed up. And so in our lab, we are about to get a preprint out on this model called Chrome BPNet, which is an extension of this original BPNet framework, where you basically first train a bias model that only predicts TN5 bias. You freeze that, and then you train a BPNet model on peaks, factoring out the bias. And it's crazy. Like, people say you can't see footprints in ATAC-seq data. But if you factor out the TN5 bias, you see extremely clean footprints. You get very clean attributions, and then from those cleaned attributions, you feed that into TF-MoDisco, and you get exactly what you'd expect. You get all the drivers of accessibility.
Do we have more questions? Are there any on the chat? Last call for questions. All right, well then let's thank our speaker again.
Thank you.